My presentation is on a study called Improvement of Migraine Symptoms with a Proprietary Supplement Containing Riboflavin, Magnesium, and CoQ10, a randomized placebo-controlled double-blind multi-center trial. Migraines are functional disorders of the brain and involve various mechanisms. Modulation of central and peripheral pain structures and release of vasoactive peptides are two of those. Migraines are often accompanied by symptoms such as nausea, phonophobia, and photophobia. The German Headache and Migraine Society and the American Academy of Neurology recommend beta blockers, anti-epileptics, and antidepressants for the treatment of migraines. In some studies, magnesium, riboflavin, and coenzyme Q10 have shown decreased levels in migraine patients. So, how are these nutrients important for migraine patients? Magnesium is a physiological antagonist at the NMDA channel involved in neuronal excitability. It's needed as a cofactor for ATP synthase, and it's needed for various processes such as vasoconstriction, platelet inhibition, secretion of serotonin, and all of these are related to the pathophysiology of migraines. Riboflavin is a precursor for FMN and FAD, which are part of complex 1 and complex 2, which are responsible for electron transport in the mitochondrial membrane. CoQ10 is a vitamin-like compound synthesized from the body from phenylalanine and tyrosine. It's needed for all cellular processes requiring energy. It is an electron carrier transferring electrons from complex 1 and complex 2 to cytochrome C. This method of treatment provides a more natural option for patients who might not be comfortable with medication or maybe just haven't responded well and are seeking out a more natural option. This combination of nutrients can be purchased commercially as a product called Dolovent in the US, and in Germany it's called Migravent. The supplement has already been tested in Germany in a clinical study with 31 migraine patients. And in this study, it was approved by the IRB for each center, and this study was registered in the German Clinical Trial Register. So for this study, this is how they found their participants and how they recruited. They looked for otherwise healthy adults of any gender, ages 18 to 65, and enrollment took place in 12 neurological centers in Germany from October 2012 to November 2013. 173 participants initially fulfilled the inclusion and exclusion criteria, but 34 fulfilled exclusion criteria after baseline and ultimately couldn't complete the study. So, in order to be included, participants must have a migraine with and without aura diagnosed according to HIS criteria ICHD 2, 1.1, and 1.2. The age at onset must be less than 50 years. Their diagnosis of migraine had to be at least 1 year before entry into this study, and they had to have at least 3 migraine attacks per month in the last 3 months, but no more than 10 headache days. They did not qualify if they used migraine prevention, such as drugs, nutritional supplements, or psychotherapy, if they have used antipsychotic or antidepressant medication in the 3 months before the study or during the study, if they have a history of overusing medication, or if they have failed to respond to 2 different prophylactic agents or are resistant to all acute migraine drugs. So the intervention, they actually called it Investigation Nutrition Product, which is a dietary food for special medical purposes according to EU regulations. It contains 400 milligrams of riboflavin, 600 milligrams of magnesium, 150 milligrams of coenzyme Q10, and then trace elements of all the other multivitamins. That combination was per 4 capsules. The trace elements included were vitamin A, vitamin C, vitamin E, thiamine, niacin, vitamin B6, vitamin B12, folic acid, vitamin D, pantothenic acid, biotin, iron, zinc, manganese, copper, chromium, molybdenum, selenium, and bioflavonoids. The placebo was indistinguishable from the INP. Patients were instructed to take 2 capsules orally in the morning and at night for 3 months, and they were only allowed to treat migraines with their usual rescue pain meds and antiemetics. The study was randomized, placebo-controlled, parallel-arm, double-blind, prospective, multi-center. After screening, participants went through a 30-day baseline period with no treatment at all, and this was to ensure more than 3 but not more than 10 headache days, and also to provide a baseline for evaluation of the other efficacy parameters. Following that baseline phase, the participants who passed inclusion criteria were randomized to verum, which is the treatment group, or placebo. The randomization was done by computer, and randomization lists were prepared. It was done by blocks of 4 per center. Both the investigator and the patient were blinded to treatments as best as possible. The treatment lasted for 3 months. Migraine parameters and intake of the investigational products was recorded daily by the patients in an online electronic diary. Compliance was monitored regularly, and they did this by reaching out if they hadn't seen the documentation for more than a week. The investigator would reach out. At the follow-up visit at the end of treatment, patients had to fill out a HIT-6 questionnaire. This measured tolerability and efficacy of the treatment from the patient's view, and they actually filled this out at the beginning of the treatment too. Concomitant meds and adverse events were checked at follow-up visits, and compliance was also assessed by pill count of the returned product. The efficacy parameters were days with migraine as recorded in the online electronic diary, and they define a migraine day as a day with at least 4 hours of migraine pain or a day with migraine pain and intake of pain medication. Maximal pain of migraine headaches, as recorded in the diary, was another parameter, and they could rate this mild, moderate, or severe. Migraine burden was another parameter assessed through the HIT-6 questionnaire. It was filled out at the start and end of treatment, and the headache impact on this scale ranges from 36, which is no headache, to 78, a very severe headache. Subjective patient evaluation of efficacy was also measured, and they could rate it very good, good, moderate, or poor. So ultimately, 34 patients could not be randomized into the treatment groups because they failed criteria after the baseline period. 4 of them had no migraine attack at all. 21 of them had between 1 and 2 per month, and 9 had more than 10 days with a migraine. 9 patients were lost during baseline due to noncompliance with the use of the online diary. 130 patients were ultimately randomized, but out of those, 1 patient provided no efficacy data since he didn't open the diary during the treatment phase and didn't show up for the final visit. And due to major protocol violations, 9 patients in the active group and 8 in the placebo group were excluded. There was no statistically significant difference in the baseline between groups. And this is a little chart to show you the baseline results. So physiological results, active treatment reduced the number of migraine days from 6.2 days to 4.4 days, but that ultimately was not statistically significant. The treatment also reduced maximal pain intensity of a migraine day based on a 3-point scale by 0.24 points, which was statistically significant compared to the placebo. The treatment reduced the sum score of HIT-6 questionnaire by 4.8 points. That took it from 61.9 points at baseline to 57.1 points after the treatment, and that was also statistically significant. And then 18% of patients in the treatment group rated efficacy of the treatment as very good, which was statistically significant compared to the placebo, but 43.9% of the placebo group rated efficacy as poor. And these are the other three tables. So safety was also monitored throughout the treatment. Adverse events were classified by system organ class and preferred term of the Med-DRA coding system. The safety population consisted of 63 patients in the treatment group and in the placebo group. There were no serious adverse events observed in the trial. The incidence of adverse events was higher in the treatment group than in the placebo group. There were 34 adverse events in 21 of 63 patients in the treatment group, which is 33.3%. And there were 9 adverse events in 7 out of 63 patients, which was 11.1% in the placebo group. All adverse events that were ultimately determined to be related to this trial were then called adverse reactions and studied further. So incidence of adverse reactions was also higher in the treatment group than in the placebo group. There were 24 adverse reactions in 15 out of 63 patients in the treatment group, which is 23.8%. And there were 3 adverse reactions in 3 out of 63 patients, which is 4.8% of the placebo group. The most frequent adverse reactions were GI disorders, and diarrhea, chromaturia were the 2 most common. Most of these were completely recovered by the end of the study. 3 of these adverse reactions resulted in dose change. 6 of them resulted in permanent discontinuation of treatment, and 2 resulted in adverse reactions to another treatment. So some weaknesses of this trial are that it was dependent on participant recording, which led to some participants ultimately not completing the trial. The patients in the treatment group could have possibly been unblinded due to a change in urine color. They warned all of the participants of this, whether they were in placebo or treatment group, to try and avoid this, but it is possible that someone was able to deduce which group they were in. In conclusion, a combination in a fixed daily dose of 600 milligrams magnesium, 400 milligrams riboflavin, and 150 milligrams CoQ10, including various other low-dose multivitamins, could reduce the intensity of migraine pain and the burden of disease caused by migraines without a high risk of serious adverse events. So this could be a good option for patients who seek a more natural method of treatment for their migraines.